So during the lecture of gastrulation, we had seen how the trilaminar germ disk is formed, how the embryo is converted into the three germ layers, the ectoderm, the endoderm and mesoderm. From this diagram, you might have perceived that the middle layer, the mesodermal layer, it is a flat layer. It is flat, undifferentiated layer. But that is not true. Actually, what happens that this middle layer, this mesoderm, this intraembryonic mesoderm, it further gets differentiated into different parts. One of the important component that it forms is called somites. So we will discuss the formation of somites as well as the differentiation of this mesoderm into different components or different parts. Hello, I am Dr. Aizaz from MedicoVisual.com. Welcome to this visual lecture. So here is the embryo and this is the ectodermal layer. If we sort of peel off this ectodermal layer, here you can see the mesoderm. So here what is this? This is the neural tube, and below this neural tube is the notochord, and here you can see that it is not a simple undifferentiated flat layer. This is the mesoderm, and you can see that it is not a simple undifferentiated layer, rather it is actually differentiated into different components. What are those components? Let's talk about that. So along with the neural tube, here is the notochord below this neural tube. This is the notochord, which is actually ventral to this neural tube. So this notochord, it is also called chordomesoderm or axial mesoderm because it is lying in the central axis, and that is why it is also called axial mesoderm. Now the part of mesoderm that is just close, which is running just parallel to this notochord or axial mesoderm, this is called paraxial mesoderm. It is running parallel to axial mesoderm, that is why it is called paraxial mesoderm. And of course it's a paired structure and it runs from cranial to caudal direction. Now in between the paraxial and lateral plate mesoderm is another component of mesoderm that is called intermediate mesoderm. So how many components of mesoderm we have studied? The paraxial mesoderm, the lateral plate mesoderm and the intermediate mesoderm. Now let's look at it in another way. Let's suppose we cut a section here. We cut a section, a cross section here, and let's see what happens. So here we have a cross sectional view of this these components of the mesoderm, and here again the neural tube, and ventral to the neural tube is the notochord or axial mesoderm, and parallel to this axial mesoderm is bilaterally the paraxial mesoderm, and of course here is the lateral plate mesoderm, here as well as here, right lateral plate as well as left lateral plate mesoderm, and you know in between them this part is the intermediate mesoderm. Now these are the components of intraembryonic mesoderm. Now what happens that, as you know, that this neural tube, especially this distal part of this neural tube, it will form the spinal cord. Spinal cord is one of the most important and delicate structures of our body. So it's very important to protect this structure. Thus, this spinal cord is protected in such a way that it is encased by the vertebral column, and this vertebral column, it is made up of small segments called vertebrae. So here there are these vertebrae, these segments called vertebrae, and they are attached together to form the vertebral column, and this vertebral column, it is encasing the spinal cord. In this way it is protecting the spinal cord. As the neural tube begins to form, this paraxial mesoderm, they take up the important task of protecting the neural tube or future spinal cord. How they do so? As you know that vertebral column is a segmented structure, so paraxial mesoderm they also become segmented, and as they become segmented they will then form the vertebral column along with the associated muscle as well as the skin of that area. These segments of paraxial mesoderm they are called somites, and this process of formation of somites from the paraxial mesoderm it is called somitogenesis. So let's first talk about the somitogenesis. These somites they are isolated condensation of embryonic mesoderm, but the cranial most part of axial mesoderm it does not take part in formation of somites. It remains partially segmented, and this cranial most part of paraxial mesoderm along with the precaudal plate and neural crest cells they will be forming head mesoderm. To understand the process of somitogenesis or formation of somites, let's zoom into this area and let's see what happens. So here is a cross section of paraxial mesoderm zoomed in, and here you can see that it consists of irregularly shaped mesenchymal cells interspersed between a ground substance. So basically, in other words, we can say that this paraxial mesoderm it is mesenchymal in nature. Here you can see that these mesenchymal cells they are far apart from each other. They are loosely arranged within the ground substance. So first of all, what happens that they come close together. As they become close together, they acquire a sort of whorled up arrangement of these mesenchymal cells. Meanwhile, incomplete ridges appear into this paraxial mesoderm, and thus this paraxial mesoderm it becomes partially segmented. It is not yet fully segmented, but it becomes partially segmented. Here in this isolated diagram you can see that this paraxial mesoderm it has become partially segmented, and each of these partial segment or block of paraxial mesoderm they are called somitomeres. So what are somitomeres? Somitomeres are condensation of whorls of mesenchymal cell into partially segmented blocks. Then what happens that these outer mesenchymal cells they undergo mesenchymal to epithelial transition, or in other words you can say that they convert into the epithelial cells. Within the center there are some mesenchymal cells which form the core, and outside are these epithelial cells. Why they convert into epithelial cells? Actually, the mesenchymal cells they do not cross-talk freely with each other, but as they convert into epithelial cells, they form the gap junction with the neighboring cells, and they sort of talk with the neighboring cells freely. What I think of it is that they are huddled up and now they are starting a meeting. So here is a cellular meeting of these cells going on. During this cellular meeting, the fate of different cells is decided. What I think of it is that the meeting is going on and they decide that hey, you four people you are going to form the dermis of skin, you four five people you are going to form the muscles of the back, and you four five remaining people you will be forming the bone of this area, the axial skeleton. And after doing so, they convert back into mesenchymal cells, and then they acquire specific fates. Some of the cell will form the dermis, some of the cell they will form the muscles, and while remaining cells they will form the bone. So let's see what happens. Meanwhile, what happens that these ridges they grow further and they completely segment these somitomeres into somites. Now they are isolated bodies, they are isolated condensations of cell, and now we call them somites. Here you can see that in the cranial part some of the somitomeres, to be precise 7 somitomeres, of course 7 are not shown here, 7 somitomeres they are not fully differentiated into somites, they are not fully segmented into somites, they remain somitomeres and they are going to form the head mesoderm. while the remaining somitomeres, they are completely segmented, they are completely transected into somites. So what is the difference between somites and somitomeres? Very simple. The somitomeres, they are partially segmented, they are not fully cut, they are partially segmented, while the somites, they are completely cut, they are completely segmented, they are isolated bodies, and that is why we call them somites. So as we have seen that the fate is decided, the fate of each of the somite is decided. So in other words, what happens that this each of the somite, it is further divided into different parts. The part that is going to form the dermis of the skin of back, it is called dermatome, while the part of somite that is going to form the muscle is called myotome, while the rest of the part of somite that will be forming the bone as well as cartilage, it is called sclerotome. So in summary, what happens that first the paraxial mesoderm is segmented into isolated bodies or condensations that are called somites, and then these somites, they are further divided into dermatome, sclerotome and myotome. Let's see in a 3D diagram. So here is the sclerotome, and the dermotome and as well as the myotome, they are closely associated with each other, so they are collectively called dermomyotome. In the cross section, sometimes it is very difficult to differentiate between the dermotome and myotome, so we can simply call it dermomyotome. Although at cellular level they are different, there is the dermotome and below that is the myotome, but collectively we can simply call it dermomyotome. So this is the dermomyotome and this is the sclerotome, and of course this is the neural tube and this is the notochord, and here is the ectoderm and here is the endoderm. Each somite, it is closely associated with the transverse segment of spinal cord or neural tube, and this is called neuromer. What is neuromer? Neuromer is the segment of neural tube. Later on we will see that from each of these segment of neural tube, from each of these neuromer, a motor as well as sensory nerve will arise, and these dermatomes and myotomes, they guide the migration of these neuronal fibers, these sensory as well as motor neuronal fibers, into the segment of dermis as well as muscles of the body respectively. And that is why what happens that in adults, the area of a skin is supplied by a particular specific sensory spinal segment. Similarly, in adults, a particular muscle group is supplied by the specific spinal segment. The area of skin that is supplied by a particular specific spinal nerve, it is called dermatome, and similarly the specific muscle group that is being supplied by a particular specific motor nerve, spinal segment of that motor nerve, it is called myotome. Now here is a confusion. In literature, what happens that the term dermatome, it is used for two things. In adult anatomy, it is said that dermatome is the area of skin that is supplied by a specific spinal segment, while in embryology you have already seen that the dermatome, it is a subsection, it is the subdivision of somite. So this can cause a confusion. So some authors suggest that we should use two different spellings. This term derma with a, dermatome, it should be reserved only for the adult anatomy, that is, it is the area of skin supplied by a specific sensory nerve, and the term dermotome with o, this term, it should be used in embryology only for the subdivision of a particular somite that forms the dermis of back. This terminology will avoid the confusion, and I totally concur with this suggestion. Although the somites form within the intraembryonic mesoderm, but they are clearly visible as small spherical elevations on the ectodermal surface of the embryo. Here you can see that these are the somites, these spherical surface elevations, the ectodermal elevations, they are called somites. First somite develop at 20th day, then the speed of development of somite is 3 somite pair per day. In human, 40 to 44 pair of somites form, and many of these somites they undergo apoptosis. They undergo programmed cell death, and only 31 to 35 somites remain. Most of these somites that have underwent the process of apoptosis, they were the part of embryonic tail, and as you know that humans do not have tail, but still there is this embryonic tail which is considered as an evolutionary vestige in humans. So because the somites in this area they are not needed, so they undergo the process of apoptosis, the programmed cell death. As we have seen that 3 somites pair formed per day, and the somites they are clearly visible as surface elevation on the ectodermal surface of the embryo. So by counting the number of somites we can roughly and vaguely estimate the age of the embryo. So this was about the paraxial mesoderm. So in summary, what happens that this paraxial mesoderm, it is ultimately segmented into somites, and these somites, each of the somite, it is further divided into dermatome, myotome and sclerotome. Then lateral to this paraxial mesoderm is the intermediate mesoderm, which forms the urogenital system that we will see later. And finally, let's talk about the lateral plate mesoderm, which is most lateral part of the intraembryonic mesoderm. Now what happens that within the center of this lateral plate mesoderm, the cell undergo apoptosis, they start dying, and small spaces appear within the lateral plate mesoderm, which ultimately coalesce to convert this lateral plate mesoderm into 2 layers. Now you can see that this lateral plate mesoderm, it has been converted into 2 layers. One layer that is continuous with the yolk sac, it is going to form the important viscera of our body, and that is why it is called visceral layer of lateral plate mesoderm. Another name of this structure is intraembryonic Splanchnopleuric mesoderm. Splanchno means it is related to viscera, it is going to form important viscera of our body, so it is called intraembryonic Splanchnopleuric mesoderm. It is continuous with the yolk sac. And then the other layer of lateral plate mesoderm that is continuous with the amniotic cavity, it is called intraembryonic Somatopleuric mesoderm. It is going to form the body wall. And in between these 2 layers is a continuous space that is called intraembryonic celom. Why it is called intraembryonic celom? Because outside whole this structure is the extraembryonic celom. Intraembryonic celom is going to form the body cavities, namely the pericardial cavity, pleural cavity as well as the peritoneal cavity.